Hello, hi, a very happy good evening. So this is Krishnaveni and we'll be talking about the story of life on earth, and that's biology. Yes, welcome to my session. So today we're going to talk about genetic disorders. So we are done with almost 3/4 of the chapter, so only the remaining 1/4 is left. So today we'll talk about genetic disorders and see if you can complete this chapter, because in this part is where the questions are a bit tricky. So I have taught you enough methods how to solve crosses. So we have solved n number of questions in crosses: monohybrid cross, dihybrid cross, incomplete dominance, codominance, etc. But now comes the most Herculean task, that is how to solve the questions from genetic disorder. If the mother is a carrier, if the father is a carrier, then how many children will be affected? So all this thing is a problem. Yes, okay, yes, hello, hi Dharani, a very happy good evening. I'm good, thank you. How are you? So please do like, share and subscribe. Okay, so welcome to my lecture on Mendelian genetics, genetic disorders. So the main agenda here is to teach you how to solve the questions and how to ensure that you have understood what is the lineage of a particular disease. So with that objective, so I move forward. So this is lecture 7 of your Mendelian genetics. So probably we'll have one more lecture on Mendelian genetics. So with that, we will come to an end of the most important chapter. So once again, an announcement. So this evening, again at 6 p.m. we are meeting. So we are talking about the important questions in biomolecules. So this lecture is at 6 p.m. So this lecture is at 6 p.m. So kindly join me. So we will discuss biomolecules. So I have to teach you one more part in biomolecules. What is feedback inhibition? What is allosteric inhibition? Okay. So that is what I have to teach in this chapter. So after which we will discuss some questions which are tricky and which are a bit expected. So please join me at 6 p.m. for this lecture. Yes, Dharni, I read your message that your 12th standard is starting officially. Yes, congratulations. So you have passed your class 11. So you asked me a method, a tip how to study efficiently. Yes, I will get back to you. Sorry, I couldn't get back when I have read your message. Hi Shimpi, yes, a very happy good evening. I'm good, thank you. How are you, Shimpi? So thank you for joining my lecture. So please do like, share and subscribe. Okay, so moving further, so this is lecture 6. Okay, so this is lecture 7, principles of inheritance and variation. So moving further, so what are the topics that we are going to discuss? So we have already discussed chromosomal theory of inheritance, genetic linkage, polygenic inheritance, what is pleiotropy, what is sex determination, and what are mutation. So these things we have already discussed. So today we move into what is genetic disorder. Okay, fine. So before we actually go into what is genetic disorder, I hope you remember what are mutations. So sudden change in the DNA sequence is your mutation. So if there is a change in a single base pair of your DNA, so this is known as point mutation. So this is known as point mutation. So when you have more than one point mutation, so this is known as gross mutation. So here we spoke about gene mutations. So gene mutations can include chromosomal mutations. So it can have deletions. So it can have insertions. It can have substitutions. So substitutions are of two types. So you have your transition and transversion, right? You have your transition and transversion, and for your deletions you have terminal deletion if it is deleted from the ends, and you have intercalary deletions. Fine, and then you have chromosomal mutations which can occur. So we are going to talk about how these mutations occur and what are the causes. Okay, so under your genetic disorders we will also talk about your chromosomal mutations. So guys, please do like the session. Yes, Swami, a very happy good evening. Thank you so much for joining my session. So please do like, share and subscribe. So chromosomal mutations include you have only one copy of a particular chromosome, so that is monosomy. So in case of Turner syndrome, you have only 45, you have only one sex chromosome, so this is your Turner syndrome. So then you have trisomy. So when you have 3 copies of a particular chromosome, that is 21st chromosome, right? So you have almost 47 chromosomes here, 23 plus 1 extra, right? So this is known as your Down syndrome. So then you have what is known as your Klinefelter's syndrome. So in your Klinefelter's syndrome, so you have XXY. So this is your Klinefelter syndrome. Okay, so we will talk about what happens in each of the cases. So I will move forward more in detail about your genetic disorder. Hi Anthony Abhi, yes, a very happy good evening. I'm good, thank you. How are you? So thank you so much for joining my lecture. So come on guys, quickly do like, share and subscribe. Okay, so moving further, so genetic disorders. So here we have this new term known as pedigree analysis. So what is a pedigree analysis and why is it important? Okay, so before we go into pedigree analysis, let me describe. Some disorders are inherited. So just like if your mother has curly hair and if you get curly hair, if these things are getting inherited, so can the disorders. So even disorders can be inherited. Okay. So in pedigree analysis, the inheritance of a particular trait is represented in the family tree over generations. Sometimes you say, it happens in the genes. Yes, so everything is in the genes. Sometimes cancer is also hereditary. Diabetes is also hereditary, right? So in these cases, yes, a pedigree analysis helps us trace back the origin. So now, if I talk about phylogeny, it is talking about ancestors, right? So in phylogeny, I have an ape mother. So ape mother gave rise to one human and one chimpanzee, right? So this gives me the evolutionary chart, or this gives me who our ancestor is. So this is my phylogenetic tree. So similarly, a pedigree analysis is a family tree which helps me trace back who will be the possibly affected offspring, who is a carrier, how can the disease expect or showcase itself in the future generation. So that is why we go forward with a pedigree chart. Okay, so if at all there is some analysis, you should know the key terms. If you have to read a graph, you should know what is x-axis, what is y-axis, right? So in that cases, there are certain symbols which you have to discuss. So directly I will show you. So this is how a pedigree chart looks like. Okay. So there are a lot of boxes which are colorful, some are not colorful. The color is of your choice. You can have any color, but here when I draw, I cannot keep coloring, but instead I will write it like this. I will shade it. I don't have to color, I can even shade it. Okay, so now what are these symbols representing? Okay, so every circle represents mother, daughter, or it's a female. So circle represents a female, okay, and a square represents a male, a son, or your father. So a male and a female, and there is a line in between. So this line signifies mating. So this line signifies mating. Okay, so the male and the female mates and they have children. So which is their first children? So you go from left to right. So this is their first daughter. So this is female, right? So daughter. So this is son. This is third son. This is okay. So this is a fourth daughter, right? Yes. So this is the order. So now this son is marrying this girl, and now they have 3 kids, first daughter, second daughter, third son. Now this girl is marrying this fellow and they are having green kids. So 3 kids. So totally everything is in violet color. So violet color is majority. Then what does red color signify here? So red color signifies it is affected. It is affected by a particular disease. So the mother is affected by a particular disease. So the second son and the fourth daughter are also affected, and thus the fourth daughter's child, that is her second daughter, is also affected. So this is how you have to ensure you read a pedigree analysis. So now going back to the symbol or the chart. So I'm moving forward. Yes, so now look. So this is male, so this represents male progeny, this is female, so this is sex unspecified. So if this is male and this is female, so this is sex unspecified. So everything is shaded or given a different color, so it means it means it is affected individual. Okay. Sandhya, Mohan, hello, hi. So I guess you're new to my lecture, so thank you for joining my lecture. So please do like, share and subscribe. Anthony, my net is slow, ma'am. I'm following you lately. That's okay, Anthony. But if you have any doubts, so please do ask me. Hi, Prince. Yes, a very happy good evening. Thank you, Prince, for joining my lecture. Guys, come on, do like, share and subscribe. Okay, if there is a line between male and female, it is called as mating. If there is a double line, what is this called? It is called as consanguineous marriages or mating between relatives. So you would have seen in earlier days, so marriages happens within the same family. They do not want to go out and give their son or daughter, so they marry within the family. So this is mating between the family members. So you have a double line. So this is called as consanguineous marriages. Okay, so that's how you pronounce. So mother, father mating, they have 2 children. So this is the first child and this is the second child. So we go from left to right. Okay. So parents with the male child is affected. Now this is colored, so which means this child is affected. So if I write like this, so this is sex unspecified. If I write 5 here, so I have 5 offsprings. It can be both male and female. Okay, so this is how I describe the symbols of a pedigree analysis. Now look into this pedigree. Okay, so now I will erase whatever I have written so that it's clear for you. So here, tell me, how do you read the pedigree analysis? So now that I have taught you the symbols, so how do you interpret this pedigree analysis? So here I have a mother, I have a father and I have the kids. Right? So this is the parental generation, this is F1 and this is F2. Right? So this is also F2. So they are of the same generation, but one is below the other one is above. Yes, Munda, hello, hi, how are you? I'm good, thank you. Thank you so much for joining my session, Munda. So please do like, share and subscribe. Okay, so pay attention. The mother is affected. That is why it is given in a different color. So the mother marries the father and 2 offsprings are affected. So when the one of the parent is affected and this offsprings are affected, so this is a dominant condition. So we already know what is the term dominant mean, right? So when one of the parent is affected and thus the offsprings are affected, in that case this pedigree is a dominant trait. Okay, it is dominant and it is autosomal. So we have sex chromosomes and we have autosomes. So autosomes are normal chromosomes, right? So which means there is a problem in the autosomal chromosome, because from the mother, if it's going to son, then you can say it is sex linked. But from the mother, it is going to the son as well as the daughter, and the mother is directly affected. So it is an autosomal dominant trait. Okay? Yes? So example is myotonic dystrophy. So please remember the example. So this is autosomal dominant. Okay? Now look at this. Look at this secondary chart. So this is our parental generation. So this is the father, this is the mother. So child 1, 2, 3, 4 and 5. So this is F1 generation, this is F2 generation. So here none of the parents are affected. So if both the parents are not affected, then it is a recessive case. But the offsprings are affected, like the son is affected, the daughter is also affected. So once again, it is an autosomal case. So the second one is autosomal recessive trait. So the example is sickle cell anemia. Okay, is this clear? So everyone watching this class understood how to interpret a pedigree analysis? Yes? Okay, ma'am, आप हिंदी बोलो ना अच्छा लगेगा? Okay, yes, I can talk Hindi when we are in an unofficial conversation, but this channel is purely English medium munna, so I cannot talk in Hindi. Okay, yes? Alright, so guys, you understood? Yes, how a pedigree analysis proceeds? Okay, fine, this is very important. You might have questions like this. Even today in class, I'm going to solve questions like this so that you will be able to interpret how to solve it. Okay, so moving further, so we are done solving pedigree analysis. Have you been to Thar Desert to look at camels? No, I have not yet been to Thar Desert. Where is lockdown going on? So when it got normal, and again the lockdown came back, so I never got a chance to visit the Thar Desert of India. So I have to visit once, but it's pretty far. So Thar Desert is not nearby, it is nearby Jaisalmer. So that's pretty far. So yes, for that I have to stop classes for 2-3 days and then go. Is that possible? No, NEET exam is nearing, right? And basically everything is closed now, so nothing is completely open. So I don't know if tourist spots are completely open. Even if I go now, I don't know if I'll be able to see the entire Thar Desert, because you have what is known as a desert resort. So that is very good, I heard. So let's see. Okay, so genetic disorders. So genetic disorders are of two types. One is Mendelian disorders, okay. Okay, so your fight is endless. Okay, now ma'am, now I am following your human physiology lectures. I will learn this later. Okay, Sandhya, but if you have any doubts, let me know immediately. Ask me, or you can join the Telegram group. The link that is given beneath my description box in my that is just below my video. Okay, Sandhya? Yes? Fine. So genetic disorders are of two types. One is Mendelian disorders, the other one is chromosomal disorders. So from the term Mendelian disorders, Mendel spoke about genes, right? So which means there are disorders which are associated with mutations on your genes. Thus your Mendelian disorders are inherited. Thus, Mendelian disorders are inherited. So, Mendelian disorders include color blindness, hemophilia, sickle cell anemia, phenylketonuria, thalassemia. So, chromosomal disorders include Down syndrome, Klinefelter's syndrome, and Turner syndrome. Okay. So first let's look about your Mendelian disorders. Let's talk about what is color blindness. So color blindness is sex-linked recessive disorder. So what do I mean by sex-linked? So sex in human beings is XY and XX. Right. So the mutation, or when I say sex-linked, it means there is a mutation in your X chromosome, because X is common to both the sexes, right? So when I say color blindness is sex-linked, there is a mutation on the X chromosome because X is common in both male and female. Okay. So it is sex-linked recessive disease. So please understand, whenever a condition known as recessive comes, so we know, suppose if A is dominant, okay, and small a is recessive. So this will express only A. So A will be expressed. So here also A will be expressed according to Mendelian law, that is monohybrid cross. And if this is recessive, only then small a will be expressed. So here in genetic disorder, there is a small twist to the story. So what is the twist? Pay attention. So it is sex-linked recessive disorder, which means the mutation is on X chromosome, right? Okay. So whenever the child or the parent has two recessive characters, then they will be deceased. Okay. Whenever they have a heterozygous condition, they will be carriers. Whenever they have homozygous dominant, they are perfectly normal. So in sex-linked recessive disorder, so females will only be carriers and males will be affected. So all the male progeny will be affected. So if you are this student who is making proper notes, so please make a note of this. So for all recessive characters or all recessive linked diseases, so for all recessive linked diseases, so please make a note. So this makes it easier when you solve questions, okay? So AA is normal. So this is my example. It can be BB, but dominant condition is normal. So heterozygous condition are carriers. They have a silent carrier, and recessive condition is they are deceased or they are affected. Okay, yes, and in case of sex-linked diseases, so always sex-linked diseases will be recessive. Here it will not be dominant, it will be recessive mostly. Rarely it will be dominant, but mostly it is recessive. So in this case, only the males are affected and the females are only carriers. So please remember these short tricks so that it is easy for you to solve. Okay? Yes? Fine. So now, what is color blindness? So color blindness is sex-linked recessive disorder, defect in either red or the green cone of the eye. So we have your cone cells which is present in your retina. If you remember the physiology of your human eye, so we have your retina, so which has your photoreceptor cells, which has pigments, so that has rods and cones. So your cones are responsible for the daylight vision and rods for the twilight vision. So you have three types of cones, red, green and blue, and a spectrum of all these gives you white light. Okay, so when you are not able to differentiate, when the stimulation between your red and the green cone is not proper, so you will not be able to distinguish between red and green color. So this is color blindness. So this is sex-linked, meaning the mutation is there in the X chromosome. Okay, so it results in the failure to discriminate between red and green color. So mutation of certain genes on the X chromosome. Fine? Yes? So Sandhya has an issue. If I touch the Telegram link, it's not coming. It's showing the page cannot be opened. So I'll ask them to refresh the Telegram link. For locomotion lesson, 2 playlists are there. One is 9 months ago, other is 3 months ago, which is advisable to follow. For locomotion lesson, 2 playlists are there. Okay, so when did I teach locomotion? So it should be in the month of February, mostly I taught. Okay, yes, the operator has given you the Telegram link, so please join Sandhya. Okay, so please follow the recent one, not the very old one. So I recently taught human physiology, so please follow them. Okay, Dharani? Yes? Fine. So now let's solve question based on this. So if the father in a family has a disease, while the mother is normal, the daughters are only inherited by this disease and not the sons. So name this type of disease. Name this type of disease. Okay, so when the father has XY chromosome, right? So mother has XX chromosome. So if they have son and daughter, so from here the gamete is only 1X. So from here the gamete is X or Y, right? So the son can be XY, the daughter can be XX. So this X and 1X is from the mother. So there is no problem with the mother, right? So if the father is having the disease and if it's directly affecting the daughter, then please understand it is sex-linked. There is the problem with your X chromosome because from the father is one X chromosome. The same X chromosome from the mother goes to the son as well as the daughter. If the son is normal but the daughter is infected, then the problem is with the X chromosome from the father. So it is sex-linked. Okay, so the answer is autosomal. Why? It is sex linked. From father to daughter it is going, so it is sex linked character. Okay, so here since the father is affected directly, the father is deceased. So the mother is normal. So since one of the parent here, okay, so this is basically you can say color blindness. So this is sex linked recessive. So mostly sex linked conditions are recessive. So here your answer is C. So here everyone understood how to solve this question. If a question of this sort comes, how do you solve? Is this clear to everyone? Yes, no doubts. Guys, come on, please do like, share and subscribe the session. So it is an important topic. Yes, this is clear to everyone how to solve? Okay. So now moving further, I have another question. So these are typical questions that comes in your NEET. So a colorblind man marries a woman, okay, with normal sight. Woman is normal, the man is colorblind. So immediately draw a pedigree chart. So you have a man, the man is colorblind. So if the man is colorblind, meaning, yes, it's a very important topic for class 12. So I write it like this. So this is the chromosome of the man. So he has color blindness. So color blindness is X-linked. So I am writing like this. Okay. So he marries a woman with a normal sight. So she is a normal one. So I write XX. Okay. Who has no history of color blindness in her family. So she is very pure. Okay. So what is the probability that the grandson being colorblind? Okay. So now this is the father, this is the mother. So assume they have two sons. Assume they have two daughters. Okay. So now here the problem is grandson. So suppose let's take one case. So this fellow marries a girl and they have a daughter, they have a son. So this daughter marries a man and she has a son. So how do you find the probability that these two grandsons are colorblind or not? Okay. So this is the condition for the parents. So if this is the gametes, the chromosomes, the gametes will be XII. From here I have only one X, right? So it will be XXI and XY. So the sons are perfectly normal in this case and the daughters are carriers. So in the F1 generation, the daughters are carriers. So the son is absolutely normal. So if he marries a normal girl, so his kids will be normal. So there is no issue. Okay, but since the daughters are carriers, so we have to be cautious. So now the daughter's chromosome is XX. So if this daughter marries a normal man, so what is the probability the grandson would be colorblind? So now I am doing the cross for this XCX, X and Y. So out of this, so this daughter is the carrier, so this daughter is normal, so this grandson is colorblind and this grandson is normal. So out of the two grandsons, so one is colorblind. So I say 50% probability that they will be colorblind. So it will be 0.5. So here we are only considering the grandson. So totally it is 100%, but since they have asked only the grandson, so this is 50, this is 50. So 50% of the grandson will be colorblind. So here it is option C. Okay, yes, all of you understood how to solve this question? Yes, you're right, you're right. Oh no, you're not right, because the sons are normal, the daughters are only carriers. Okay, Prince? Yes, ma'am. Oh, very happy good evening. Thank you so much for joining my lecture. So please do like, share and subscribe. So I hope this is clear. Okay, so this is about color blindness. Now the next topic is hemophilia. So this is also sex-linked recessive disease. So if you are making notes, write down color blindness, sex-linked recessive. Then hemophilia, this is also sex-linked diseases. So this is transmission from unaffected carrier female to some of the male progeny. So it is very similar to your color blindness. So the females will be only carriers and the males will be the affected progeny. So what happens in case of hemophilia? So in your color blindness, it was that your cones are not stimulating properly. So what happens in your hemophilia? So the protein involved in the cascade of blood clot is affected. So you have clotting factors, right? So your blood has to clot. So if you get hurt, your blood will come out, your blood will ooze out, but very soon it has to stop. So you do not have excess bleeding. So whenever you have fallen from the cycle, yes, it bleeds, but quickly the bleeding stops, because you have clotting factors that is induced by your platelets. If at all you do not have this clotting process happening, you will lose a lot of blood. So this clotting factor is absent when a particular protein is not coded. So the gene is deleted. So that is the disease known as hemophilia. So you have this entire process for clotting, which finally creates blood clot. When even one of the protein is missing, then the blood will not clot. So this is your hemophilia. So you will have non-stop bleeding. So we all know Queen Victoria of UK. So she has been the carrier of hemophilia. So all the male progeny in their family history, they are suffering from hemophilia. Okay, so this is a well-known case. Fine. So now I have a question based on hemophilia. Okay, so before that we move to sickle cell anemia. So I will make it clear. So I have spoken about sex-linked recessive diseases. So what are they? Sex-linked recessive. First one is color blindness. The next one is hemophilia. Right? So now we are moving into the next part of the disease. So what is the next part of the disease? Sickle cell anemia. So it is autosome linked recessive disease. So for all these cases, so we had a mutation in the X chromosome. So here the females were only carriers. So here the male progeny was affected. But now we are moving into a next category of autosomal diseases. So which means their sex chromosomes are normal, mutation happens in the autosome. So what happens in the autosome? So first example is your sickle cell anemia. So we know what happens in case of your sickle cell anemia. So your RBC becomes sickle cell shaped. So it becomes the shape of your sickle because you have a point mutation from GUG to GAG. So that is why sickle cell anemia happens, but it is autosomal disease. So it is autosomal recessive disease. So autosomal means sex chromosome, autosome means non-sex chromosome. Recessive means both the parents need not be affected. So the condition can be the father is not affected, the mother is not affected, but the child can be affected. Maybe they are carriers. So if at all the son is like this, then he is affected. So this is autosomal recessive. So the parents are silent carriers of this gene. So this is your sickle cell anemia. Okay, so moving forward, so it is autosomal linked recessive when both the parents are carriers for the gene. So normal condition, if a person has normal RBC, so he will be HBA and he will be HBA. He will have this pair of allele. If suppose a parent is a carrier, then he will be HBA and HBS. So if a person is having sickle cell anemia, so this is HBS, HBS. So it will be a recessive condition. So this is the deceased person. So he is the carrier. he is normal. Okay, so your RBC becomes sickle cell shaped. So one more okay, so till this you remember. So the next one is phenylketonuria. Yes, questions are there, it's coming at the back. So phenylketonuria, you know what is S. So S stands for sickle cell anemia. So here you have yes, so A is normal, okay, so we are just assuming it to be HbA normal, and S is your sickle cell shaped, okay, it's an allele which codes for your sickle cell protein. So the next one is phenylketonuria. So we have read this n number of times. This is an example of pleiotropic gene. So when your amino acid, that is your phenylalanine, gets converted into tyrosine, that is the only way this can be metabolized. So for this we need an enzyme, phenylalanin hydroxylase. So if this enzyme is deleted, then this conversion is halted. So accumulation of phenylalanin causes phenylketonuria. So it lacks an enzyme, but this is also an autosomal recessive trait. Okay, so this is completely normal. So here it is carriers. So same condition as sickle cell anemia, and they are affected. Since these are inherited, you have conditions like this. Okay, so this causes mental retardation. So this is how these kids who are suffering from phenylketonuria look like. Okay, so now the last disease in your Mendelian genetics, that is your thalassemia. So autosome linked recessive blood disease which is transmitted. So it is due to mutation or deletion. So in your hemoglobin you have 4 subunits, right? So you have 2 alpha subunits and 2 beta subunits. So when you have a mutation in the alpha subunit, you call it as thalassemia alpha, but when you have a mutation in the beta subunit, you call it as thalassemia beta. Okay, so HbA1 and HbA2, so 2 alpha subunits are in the 16th chromosome, whereas the beta chromosomes are on the 11. So please remember 11 and 16. So if you have thalassemia alpha, so mutation is in the alpha subunit of your hemoglobin, so that is on chromosome number 16. Okay, yes, so thalassemia is again related to your hemoglobin structure. Okay, yes, fine. So now here comes the question. Yes, Arshita, after a long time you are here. Hello, yes, a very happy good evening. I'm good. How are you? So guys, come on, please do like the session at least. So I know you're all very busy, but I don't think you're too busy to even not like the session. So please do like the sessions. Please lend us your support. Okay, so now let's solve interesting questions. So if both the parents are carriers of thalassemia, so here thalassemia is autosomal recessive. Sometimes it will be mentioned in the question. So what is the particular type of disease? Sickle cell anemia is autosomal recessive. Color blindness is sex-linked recessive. Right? So thalassemia is autosomal recessive disease. What are the chances of pregnancy resulting in an affected child? So here they have given both the parents are carriers. So I draw the parent, the father, the mother. So both the parents are carriers, so this is the condition. Okay, so now if suppose they have 4 children, so what is the probability or chances that the offspring is affected? Meaning the offspring has to suffer, has to be affected by thalassemia. So this is the gamete. So now tell me what is the answer. So this fellow is completely normal. So this fellow is completely normal. So these two are carriers and this one is affected. So out of 4 kids, so 1/4 is affected. So that is 0.25. So you understood how to solve this? Yes. So now the answer is 25%. So you understood how to solve? Yes. So this is normal. These two are carriers of thalassemia and this one fellow is affected. It can be male or female, it does not matter because it is an autosomal disease. It can affect anyone, right? So here 1/4 is the answer. So please take a minute to see if you have understood or not. Yes, Shubham sir, hello, very happy good evening. So thank you so much for joining my lecture. So please do like, share and subscribe. Yes, everyone understood, Prince? Clear, everyone? So can I move forward? Okay, so moving forward. So now comes chromosomal disorders. Yes, once I finish chromosomal disorders, I have inline 15 questions. So if you're not able to solve the 15 questions, you will solve it as homework and come. So that includes your pedigree analysis, your questions from Mendelian genetics as well. Okay, so chromosomal disorders, so absence or abnormal arrangement of one or more chromosome. So aneuploidy is gain or loss of chromosome. Polyploidy is increase in the set of the whole chromosome. So aneuploidy has 3 types. So aneuploidy. So always ploidy is chromosome number. Aneuploidy, so this can be of 2 types. Either you can have a loss of chromosome. In case of monosomy, you have only 1 pair of a chromosome. So you can have a gain of chromosome, that is trisomy. So ideally it should be 2. So when you have 3, it is gain. When you have 1, it is loss, right? So the example of monosomy is Turner's syndrome. So Turner's syndrome is in case of females, okay. So this mostly is in case of females. So females have 1 sex chromosome less. So they have XII condition. So they have only 45 chromosomes. Yes sir, a very happy good evening. So please do like, share and subscribe. Okay. So here for trisomy, the example is Down syndrome. So Down syndrome is mostly in case of, it can be a boy or girl, it can be male or female, but you have extra copy of 21 chromosomes. You have 3 copy of this. So this is your aneuploidy. Okay, yes, clear? So Down syndrome is 3 copies of your 21st chromosome. You also have another example for your trisomy. So that is your KLINEFELTER'S syndrome. So they also have 3 sex chromosomes, that is XXY. So KLINEFELTER'S syndrome is mostly in male. So they have 3 sex chromosomes. So totally they have 47 chromosomes. Okay. Yes, I will show you the condition now. So I have explained. So Down syndrome is additional copy of 21st chromosome. So it is trisomy. So it was described by a person known as Langdon Down. So fusion of a normal sperm with an abnormal egg. So that leads to this problem. So it arises due to chromosomal non-disjunction. So if I have 2 chromosomes, right, so this is 21st chromosome, this is 21st chromosome. So in case of the egg, so if I have to form the egg, so only one of this should come down, right, but both comes down. So here the pairing or the random segregation is not proper. So this is a person or a child with Down syndrome. So Down syndrome person or child will not have high mortality. They usually die between 3 or 4 years. Then KLINEFELTER'S and Turner's syndrome. KLINEFELTER'S is when you have additional copy of X chromosome. So this, since they have a Y chromosome here, so they are mostly male, but they have female characteristics as well. So the female characteristic is the development of breast. So when a male has the development of breast, so that condition is known as gynecomastia, gynecology. So gynecomastia is the development of breast in male. So this is basically your transgenders. Then they are basically sterile, they cannot reproduce. Then Turner's syndrome is absence of 1 X chromosome. You have only 45 chromosomes here. So such females are sterile. Okay, so this is known as karyotyping. So I have taught you what is karyotyping. So in the chapter Cell, the Unit of Life, so where you study the structure of chromosome, the shapes of chromosome, the number of chromosome, if you look, so you have 2 X chromosome. So this is how they find out it is a Klinefelter's syndrome. Yes, so this overall masculine development breast, it happens when they cross puberty. So when in the embryo stage, if you have to find out, you do karyotyping, you take a piece of your amniotic tissue and study. All right, yes. Okay, so with this we come to an end of your chapter, that is Principles of Inheritance and Variation. Now what is remaining is to solve questions from this portion. So let's see if you can solve this for me. So which of the following pair is wrongly matched? Yes. Yes, Anthony, you're right. So which of the following is wrongly matched? So if you understand, it is a NEET 2018 question. XO's type of sex determination is seen in grasshopper. So I have already taught you how sex determination happens. So in case of insects, it is SO. So humans, it is XY. In birds, it is ZW. So in birds, it is female who determine the sex. In males, in humans and in insects, it is males. So ABO blood grouping is codominance. Starch synthesis in P is multiple allelism. TH Morgan found linkage. So which is wrongly matched? Yes, this is not multiple linkage. This is this is incomplete dominance, right? So option 3 is wrongly matched. Yes, right. Okay, so moving further. So question 2, I'm sorry. Male heterogamity is seen in humans, grasshopper, Drosophila, all of these. So male heterogamity is when the male sex chromosome has heterozygous condition. It is known as male heterozygosity. Now here the male determines the sex of the offspring. So male heterogamity is seen in all of these. Yes, absolutely right. So in humans, it is XY. In grasshopper and Drosophila, it is XO. Right? Yes. Oh, Shrimpi, this is your favorite answer. Okay. So question number 3. The number of chromosomes in females and males of honeybees are. So in honeybees, we have haplodiploid system, right? So the males are haploid and the females are diploid. So the males are haploid and the females are diploid. Yes, if the females are 32 and the males should be 16. You do not have to know honeybee has 32 chromosomes. You just have to know males are 2n and females are 2n and males are n. So you can easily arrive at the answer. Okay? Yes. So question 4, see if you can understand. If there are 4 different types of nitrogenous bases in your DNA, ATGC, then how many different types of transitions and transversions are possible? So transition and transversion are type of substitution. If a purine is substituted with a purine, it is transition. If a pyrimidine is substituted with a pyrimidine, it is transition. So transversion is when a purine is substituted with a pyrimidine. Okay, so your purines include adenine, guanine. Your pyrimidines include cytosine and thymine. So how many purines and how many transitions and transversions are possible? Yes? Yes, question 4. So please think and give me the answer. Yes, Prince, you're absolutely right. So transition is 8, transversion is only 4. So why 4? So transition is when A can be converted into G. So G can be converted into A. So C to T and T to C. Right? So it can take any form. So transversion is only 4. So this is 1, 2, 3, 4. So into 2 times. So it can have 2 types. So this is purine with a purine, pyrimidine with a pyrimidine. So that is transition, right? Yes. Option A and C are the same. Transition and transversion. So I guess I have, yes, option A and C are same. So yes, it is right. So transversion is only A can be converted into T or G. Okay, 1, 2, and G can be converted into C or T. Right? So this is C. So this is 4 and this is 8. Yes, did everyone understand? So transition is a purine replaced by a purine, a pyrimidine replaced by a pyrimidine. Whereas transversion is a purine replaced by a pyrimidine and a pyrimidine replaced by a purine. Yes, do you understand? Yes. So I will check option A and C once again, and I will get this question in the next class as well. So now let's move forward. So I have to check question number 4. I will remember. So question 5. Frame shift mutation arises due to deletion of base pair of DNA, insertion of a base pair of DNA, both A and B, change in a single base pair of DNA. So change in a single base pair of DNA is your point mutation. So D is not the right answer. D is not the right answer, Prince. So frame shift mutation is change in the reading frame. So when I write the hat is red, the hat was red. So now it is easy for you to read. So if at all, so my proteins are read in triplet codons. If at all there is a deletion here, so then it becomes like this, right? So this changes my reading frame. So deletion is the cause. So if I have an insertion, so if I insert an I here, so it is THE, IHA, then TWA, HRE, then D. So both your deletion and insertion causes change in the reading frame, that is your frame shift mutation. So question number 6. So identify the correct choice for the given symbols. So if this is from your pedigree, so this is your male, this is your female. So this is your male, this is your female. So if you have one line, it is mating. So if you have 2 lines, I told you it is marriage or mating within relatives, that is consanguineous marriages. So consanguineous marriages are a bit risky because recessive genes tend to get expressed. Alright, so here what is the right answer? So here what is the right answer? So question number 6. Yes, both B and C are right. So A is mating. So B is mating between relatives or consanguineous mating. Yes, absolutely right. Very good, Prince. Okay. Fine. So moving further, yes, I will stop with this. So you solve the remaining 8 questions as homework. So this will be my last question. So following pedigree chart shows which of the following. So this is the mother. So this is the parental generation. So this is the mother. So this is the father. So here, so this is their child, first child, second child, third child, fourth child. So this is F1, this is F2, and in F2 also the sons are only affected. So here the father is affected, then the sons are affected. So the character is sex linked, which one? Yes, it is sex linked. Okay, so here it is dominant because one parent is affected, right? So it is very clear, if one parent is affected, then it is a dominant condition. But from father, it is only moving to sons. It is not moving to daughter, right? So it cannot be a recessive character. So it has to be a dominant character. So the character is carried by, okay, so here you can say, here the character is sex-linked dominant. since from the father it is going only to the sons. So you call such characters which are passed only by your X chromosome. Okay, so you can also say it is a character carried by Y chromosome because only the male progeny is getting affected. So those characters are called as Hollandric characters. Okay, but here since one parent is affected, so according to our pedigree introduction, so it is a dominant condition. So I say this character is sex-linked dominant. Okay, yes. Is this clear, everyone? So I stop here, but I still have 8 more questions which you will do as homework and come. Okay, yes, so you have a lot of pedigree based questions, so please do them and come. In the next class we will discuss. So the next class for genetics will happen on Friday. Okay, yes, so please solve them and don't forget to join me for my 6 o'clock session. So there also we are discussing questions from biomolecules. So please join me. So if you genuinely like my way of teaching, so please do like, share and subscribe, and thank you so much for joining my lecture today. So please do join me at 6 p.m. as well. So this is me wishing you more and more. So take care and bye-bye.